All right. So when you're thinking about these genetic defects, think about this whole path only works for a genetic defect, right? So there's some inheritance pattern. You want to know that. Is it autosomal recessive or dominant? What is the mutation? Because the mutation can make a difference in the therapy you give. Okay, so what happens normally? You have DNA, you transcribe it, you make RNA, that's spliced, that's translated into protein. That protein goes on and does something. It can be inhibited by something, by an inhibitor. If it works, the enzyme takes some reactant to some product. Okay, this is the generic view of what's going on with proteins. And at each of these steps, if it's not working, you can have a therapy. If this is the problem at the DNA level, gene therapy is the thing you want. We're working on a bunch of those. In 15 years, we should have a very different view. A lot should be done, but we're not there. Read-through inhibitors are a kind of really important thing that are not talked about a lot. I'll mention them more later, but in long story short, if you have I think I mentioned this in the protein lecture, if you have a nonsense mutation, if you have a mutation that causes a stop codon too early, let's say you're supposed to be 300 amino acids and it stops at 50, that's going to go off and be degraded. A read-through inhibitor allows the molecular machinery to pass by that like 10% of the time. So it gets past the one mutation, keeps going to the next stop, which is the correct, and then you get enough protein to not have a phenotype. Amazingly, that means you're going past the normal stop codon in all your other genes 10% of the time, but the read-through inhibitors are only used for diseases where your life expectancy is lowered. So any hit of those, I've tried to look up side effects of read-through inhibitors, and they seem to be low enough that there's a benefit whenever people use them. Again, think if you read through the stop codon on a normal one, usually that's way too big, and that's going to end up being degraded anyway. So you're kind of probably just using a little too much ATP. Okay. There are drugs starting to try to aim at splicing, but they're not really doing a lot yet. But that is another place you could go where if you have a genetic defect at one of these points that causes a splice problem, you could potentially correct that and get the right protein. There's small molecules that help chaperone, so they help proteins fold. Cystic fibrosis is the poster child of that. You can have specific mutations that just make your CFTR not quite fold correctly, and if you can add a drug that makes it fold well enough, it gets into the membrane and you're fine. And then you could do substrate reduction. So if the problem is that this enzyme isn't working well enough, you could try to reduce the reactant so that's not building up, right? If this green Pac-Man isn't working well, the problem is often a buildup of this reactant. Now that won't help if the problem is not getting the product, right? And then enzyme replacement therapy, that is dependent on delivery mechanism where the enzyme needs to be, right? If it's a digestive problem, that's great. If it's something in the brain, how are you going to do that? Does that make sense to everyone, generically?